Welcome to Lecture 16, where we will explore the immune system. Immune cells have the ability to recognize foreign pathogens. They can do this innately by recognizing structures or molecules that are not present in animal cells, or they can do so adaptively through the prior exposure to pathogens. In innate recognition, each of the small set of receptors can recognize a molecule that is absent from animals, but common to a type of pathogen, such as a virus, bacterium, or fungus. Innate immunity is common to all animals. However, adaptive immunity is only found in certain vertebrates. In adaptive immunity or adaptive recognition, there will exist a vast number of receptors that are specific for a particular part of a protein on a pathogen. An example is the body being able to detect a surface protein of the influenza virus. In this example, as seen below, a different adaptive immune receptor would be present within the body to recognize each of the flu strains based on their protein amino acid sequence. Note that each of the strains differs at the positions that are colored in orange. Recognition will then trigger a response to help protect the infected animal. We are now going to take a look at innate immunity, particularly in invertebrates. Many animals have physical barriers that help to prevent infection. These can include the exoskeletons of arthropods, as well as the internal linings of the various types of systems that help to protect the body from infection. However, if these physical barriers are breached, recognition proteins can recognize molecules that are not present within the individual organism. These recognition proteins include lysozymes and hemocytes. Lysozymes are present in the digestive system and can help break down bacterial cell walls if bacteria is ingested within the food particles. Hemocytes, present in insects, can help phagocytize pathogens and may also release antimicrobial peptides that help to target bacterial cell membranes. For example, if a fungus is ingested along with a food item, the binding of recognition proteins will bind to the fungal cell wall. This will activate a transmembrane receptor called Toll. This will then activate the production and secretion of antifungal agents to destroy the fungal pathogen. This diagram depicts the ingestion and destruction of pathogens in general. A phagocytic cell will extend out pseudopodia or false feet that will surround the pathogen. This will then cause the pathogen to become engulfed by the process of endocytosis. Once the pathogen is brought in, a vacuole will form around the ingested material. This vacuole will then fuse with a lysosome. This will then release hydrolytic enzymes into the vacuole containing the pathogens. This will then lead to the destruction of the pathogens. The debris from the digested pathogens will then be released into the environment. We are now going to take a look at innate immunity in vertebrates. Vertebrates also have physical barriers that help to prevent infection. For instance, humans have skin as well as mucous membranes that help block pathogen entry into the body. In addition, body secretions such as tears, saliva, and mucus can also create inhospitable environments for pathogens. These secretions may also have lysosomes to help destroy pathogens if they come into contact. Mammals also have various toll-like receptors, or TLRs, on and in phagocytic cells. These TLRs can help recognize specific pathogenic material or foreign material. Take a look at the diagram below to see the role of various types of TLRs in the identification of lipopolysaccharides, particular DNA or RNA sequences, as well as flagellin. Binding of these toll-like receptors will trigger an immune response that can include cytokine production as well as the secretion of antimicrobial peptides. In general, within mammals, when we are discussing innate immunity, we can look at the action of two main types of phagocytic cells that will help to recognize foreign pathogens: neutrophils and macrophages. Neutrophils will travel into the blood to infected tissues to help engulf and destroy pathogens. Macrophages are larger phagocytic cells. Some may travel within the body, however, many are localized within organs and tissues and provide a first line of defense for infectious particles or pathogens. In addition, within mammals, there are also many other immune cells, such as dendritic cells that are located in the skin or high contact tissues. Dendritic cells are also involved in adaptive immunity after a pathogen is engulfed by the cell. Thus, they are involved in both innate and acquired immunity. Eosinophils are located under the epithelium. These cells help to discharge enzymes whenever pathogens or parasites are encountered. NK, or natural killer cells, circulate in the body and help to detect any abnormal surface proteins that are present on infected or cancerous cells. Once these abnormal surface proteins are detected by the NK cells, this will cause the release of apoptosis or cell-killing inducing chemicals. Also note, NK cells can also be involved in adaptive immunity. And finally, mast cells are present in connective tissues. Mast cells are very involved in an inflammation or immune response when allergens are encountered. We are now going to take a look at various types of inflammatory or immune responses, first beginning with a local inflammatory response. This type of response is initiated whenever there is injury or a primary site of infection. In this type of response, macrophages will release cytokines. The release of these cytokines helps to recruit neutrophils to the area of concern. Mast cells are also involved, and they will release histamine. Histamine will cause surrounding blood vessels to dilate. This causes the initial area of infection or of concern to become warm and flushed. If the signaling is prolonged, this will recruit more phagocytic cells to the area, as well as increased blood flow. The increased blood flow will also bring antimicrobial peptides to help kill the pathogens. As the pathogens are being killed off and the white blood cells are becoming active, this will produce pus. Pus in general consists of white blood cells, pathogens, as well as any surrounding tissue or cellular debris. This pus will then enter into the lymphatic system, and any exposed dendritic cells will then migrate into the lymph nodes. This can then initiate an adaptive or acquired immunity response. Take a look at the diagram to understand the different cells involved in the local inflammatory response. We have the mast cells secreting histamine, we have macrophages releasing cytokines, dilation of the blood vessels, and eventually recruitment of neutrophils into the area. The interstitial fluid, which is present at the inflammation site, as well as the white blood cells, will eventually enter into the lymphatic vessels. This lymph will then be brought to the lymph nodes, where macrophages and other defense cells are further activated. Take a look at the diagram to understand the relationship between interstitial fluid and the lymph system, as well as the overall location of the major lymph vessels, as well as the aggregation of lymph nodes. We're now going to take a look at what occurs in systemic or a chronic inflammatory response. If there is extensive injury in a particular area or extensive infection, this may lead to a more systemic-based response as opposed to a local response. In this instance, macrophages will release pyrogenic cytokines. This will help to reset the body's thermostat, which causes an overall rise in body temperature, which is also considered to be a fever. This can serve to increase the immune response, rate of tissue repair, and also aid in killing the infective pathogen. If body cells are infected with a virus, those infected body cells may release interferons. These are chemical signals that will help to prevent viral replication to help limit the spread of the virus in the body. In addition, multiple inactive proteins help to make up the complement system. This may become activated by substances on surfaces of many pathogens. This will also help to initiate an immune response and also will cause the lysing of any infected cells. However, some infections may become problematic as they may lead to prolonged high fevers, low blood pressure, poor blood flow, and a potentially fatal condition called sepsis, or toxic shock. Chronic conditions, such as colitis and Crohn's disease, can also be very damaging to human health. In addition, some pathogens have evolved the ability to evade detection, or they can resist degradation after ingestion by macrophages. For instance, some bacteria, like the Streptococcus pneumoniae, has an outer capsule that helps to aid in evading a host's immune system. As a result, these infections can be very persistent, resistant to many different treatments, and can become deadly if left uncontrolled. We are now going to take a look at what occurs in adaptive or acquired immunity. Adaptive immunity responses are a unique adaptation in vertebrates. It involves the specific recognition of molecules or antigens on a pathogen through the use of antigen receptors on antibodies or specific immune cells. Antigens will bind to receptors at specific regions called the epitope. Immunity against a pathogen can increase once the organism has been previously exposed to that pathogen. Adaptive or acquired immunity involves two major cell types, T cells and B cells. T cells are immune cells that mature in the thymus gland. These cells can help bind to antigens or fragments of the antigen that are attached to an infected host cell through the use of an MHC or major histocompatibility complex molecule. B cells, on the other hand, mature in the bone marrow. B cells have Y-shaped antigen receptors that once they bind to an antigen activate the cell to secrete antibodies or immunoglobulins. Remember, both B cells and C cells originate from stem cells that are present in the bone marrow. Take a look at the diagram to the right to understand the structural differences between the antigen receptors on B cells versus T cells. Receptors on the B cells have both heavy and light protein chains that are kept together by disulfide bridges. Receptors on the B cells also have multiple binding sites for the antigen. Receptors on T cells, on the other hand, have alpha and beta chains that are held together by a disulfide bridge. These will combine to form the antigen binding site. Also note that B cells and T cells differ with respect to their mode of action. T cells attach to antigens or fragments of the antigens that are attached to MHC molecules or major histocompatibility complex molecules that are present on infected host cells. Thus, they have to have cellular contact. B cells, on the other hand, can attach to either free or bound antigens. Thus, they do not necessarily have to have cell-to-cell contact. They can also interact with the free-floating antigens that are circulating within an area. There are many important changes that occur in the development or maturation of B and T cells. As a lymphocyte matures, they can develop receptor diversity, meaning that they have the ability to detect antigens and pathogens despite not having any previous exposure to the antigen. And they also have the ability to be self-tolerant, which means that they do not react to the host's own cells. As a mature lymphocyte encounters more antigens, they also develop two other aspects as they mature, the ability to proliferate and the ability to have memory. When a lymphocyte matures, they have the ability to rapidly increase the number of B and T cells that react to a specific antigen. Thus, by undergoing cell proliferation, they can increase the number of immune cells that are responsible for detecting specific antigens. They also have immunological memory, which means that their response to the antigen gets faster and stronger upon each exposure. Also note that we have a difference between the primary immune response, which occurs upon initial exposure to an antigen, versus a secondary immune response. In a primary immune response, the peak response or immune response in the body occurs approximately 10 to 17 days after the first exposure to the pathogen. However, if a body is already exposed to this pathogen and we have a secondary immune response, the immune response can actually peak much quicker, peaking within 2 to 7 days after initial exposure. This is due to cell proliferation as well as the concept of immunological memory. Lymphocytes with the antigen will travel and then get trapped in the lymph nodes. They will reside here until a match is made, meaning that the antigen needs to bind to the correct epitope on the correct lymphocyte. This is called clonal selection. Once the T or B cell is activated, it will undergo multiple rounds of cell division to produce clones. These clones will either become effector cells or memory cells. Effector cells are considered to be short-lived cells that can take immediate action against any antigen or infected cell. Beta cells can produce plasma cells, whereas T cells can produce helper T cells. Memory cells, on the other hand, are longer-lived cells. They can also produce effector cells if the antigen is encountered at any other point in time. This diagram depicts the difference between primary and secondary response and also shows the specificity of immune responses. If we take a look at the graph below, we can see that on days 7 to 14, we see an increase in the production of antibodies to antigen A. This is involved in the primary immune response. Once this drops off, if there is a secondary exposure to antigen A, we can see that the immune response occurs quicker as well as to a much higher degree, meaning that many more antibodies are produced against the pathogen or against the antigen than were produced in the initial primary exposure. Also note that even though we have a acquired and a quicker secondary immune response to antigen A, this does not provide us immunity against antigen B. Thus, every single exposure to a different antigen will lead to an independent immune response.